Hello everyone. In our previous lecture, we talked about metabolism in general, but in this lecture today, we will look at the process of sugar splitting, which is also known as glycolysis. There are three major pathways to completely oxidize a sugar molecule. So in the first stage, you have glycolysis, or the breaking down of a sugar molecule into smaller units, such as pyruvate and acetyl coenzyme A. Then acetyl coenzyme A will enter into the citric acid cycle that produces carbon dioxide and many high energy electron carrier molecules such as NADH and FADH2. And eventually, those energy trapped in the electron carriers will be used in generating ATP in the oxidative phosphorylation that happens right in the mitochondria. So these are the learning outcomes for this session. We'll first look at what is glycolysis and the purpose of glycolysis and why is it so important to all living things. And then you should be able to outline the 3 stages and 10 steps in glycolysis and to know the enzymes required in each of the reaction. And last but not least, you will know the roles and importance of NAD positive in glycolysis. So glyco means sweet while lysis means splitting. So essentially, glycolysis is the splitting of sugar into smaller molecules for energy production. Due to glycolysis, carbohydrates can be used as a fuel molecule in living organism. But carbohydrates first have to be hydrolyzed by different enzymes into glucose molecule, which is the basic monomer for all carbohydrates. And eventually, glucose molecule is going to be converted into pyruvate, which is the end product of glycolysis. So there are many scientists who discovered the process of glycolysis, but mainly you have two scientists who are Emden and Meyerhof. And for this reason, glycolysis is also known as the Emden-Meyerhof pathway. So one important note about glycolysis is that it happens right in the cytoplasm, not in mitochondria. As a result, can prokaryotes undergo glycolysis? Yes, because they do have cytoplasm. So although they don't have mitochondria, they don't have any organelles, but because glycolysis happens in the cytoplasm, so all prokaryotes can undergo glycolysis. And a second thing is that glycolysis is an anaerobic process that doesn't require oxygen presence. So why is glycolysis important? Because it is the only pathway in the body that can generate energy without oxygen. As I mentioned, glycolysis is an anaerobic reaction, and it does so by converting glucose into pyruvate molecule. Essentially, glycolysis can be broken down into 3 stages with 10 different steps or 10 different reactions. Stage 1 is known as the investment or the priming stage, where investment of ATP is required to convert glucose into fructose 1,6-bisphosphate. Then in stage 2, you have the splitting of fructose 1,6-bisphosphate, which is a 6-carbon sugar, into two 3-carbon sugar, which is the glucose 3-phosphate molecule. And in the final stage, you have the energy yield or the energy conservation stage, by which G3P is converted into pyruvate molecule. So one point to take note is that in glycolysis, only a portion of energy in glucose is released as ATP, while we have more energy stored in this molecule of pyruvate, which can be oxidized further. And now we will look at glycolysis with more details, and you can actually find this similar diagram in your textbook. So first of all, you don't have to memorize everything, such as the structures in a pathway, but you are expected to remember the names of the metabolites and the enzymes in the process of glycolysis. So in order to release energy from glucose molecule, we'll first have to invest energy into this stage 1 of glycolysis process. This is very similar to financial investment, where we have to invest a small amount of money before we can earn more money in future. So remember, in stage 1 of glycolysis, our body invested a small amount of ATP so that we can harvest even more ATPs later in stage 3 of glycolysis. Therefore, ATP is being consumed rather than being produced in stage 1 of glycolysis. Now, if you look at the first stage of glycolysis, ATP is invested 2 times. And whenever an ATP is being invested, you have the phosphorylation of the metabolite molecule from glucose into glucose 6-phosphate and from fructose 6-phosphate into fructose 1,6-bisphosphate. That's because the phosphate group has been transferred from the ATP to the metabolite molecule. So why do we need phosphorylation of the substrate molecule? I will explain this in the next slide. Well, we know that glucose are circulated in our bloodstream and they need to be imported inside the cell. But when you have too much glucose in your blood, that is not good for the body, and it is known as hyperglycemia because it can cause a lot of damage to your body and organs. So due to high sugar level, some of the glucose is going to be urinated out of your body, and this can hurt your kidney, and it is a waste of energy as glucose is removed from your body. So it is preferable to make glucose stay within the cell. But remember, glucose can diffuse in or out of the cell through diffusion by using this glucose transporter molecule. Therefore, glucose will only move according to the concentration gradient and arrives at the equilibrium. So, how can we pump more glucose into the cell? Well, the strategy is to convert glucose into glucose 6-phosphate. Due to the presence of this phosphate group, the glucose 6-phosphate is no longer recognized by the glucose transporter, and hence they cannot escape the cell. And a second benefit is that with the addition of the phosphate group, this stabilizes the whole G6P molecule, making it more reactive for subsequent reactions. So this reaction is catalyzed by the enzyme hexokinase, which has a very low Km value of 0.1 millimole per liter. So when the Km is low, it means that this enzyme has a very high substrate affinity. So as soon as a glucose molecule enters the cell, they will be bound with the hexokinase enzyme and be converted right into G6P. So this reduces the amount of glucose within the cell, and it further promotes the equilibrium towards the import of glucose into the cell. Okay, the enzyme catalyzing this first step is known as hexokinase. And when you see the term kinase, it is always associated with ATP. So what ATP does is to transfer one phosphate group to the C6 or carbon 6 of the glucose molecule, forming up glucose 6-phosphate, while ATP has now become ADP. So this enzyme will require a cofactor to function optimally, which is a magnesium 2 positive ion. So why is it Mg2 positive? Now here is a plausible explanation. So this is an ATP linked up with 3 negatively charged phosphate groups. But in this process, we will only want to release one phosphate group. So it is logical how the presence of this divalent 2 positive magnesium ion can stabilize the 2 phosphate groups by neutralizing the negative charges on these 2 phosphate groups. So to recap, in step 1, we have the conversion of glucose into glucose 6-phosphate molecule by the action of hexokinase. And this enzyme will require the presence of ATP and the cofactor Mg2 positive to function optimally. And the function of ATP is to transfer a phosphate group to the carbon 6 of a glucose molecule, forming this glucose 6-phosphate molecule, while both ADP and the magnesium 2 positive ions can be recycled. Then we come to step 2, where the glucose 6-phosphate is converted into fructose 6-phosphate by the enzyme phosphoglucose isomerase. So what isomerase does is to rearrange the atoms in the molecule to produce the isomer of G6P, which is F6P in this case. Now in fact, G6P is an aldopyranose, which is a 6-membered ring structure with an aldehyde group as the functional group, while F6P is a ketofuranose, which is a 5-membered ring structure with a ketone functional group. Now in short, in step 2, you have the isomerization of G6P into F6P. So to catalyze this reaction, the enzyme needs to first open up the ring structure exposing the aldehyde functional group of the G6P. Then it catalyzes the transfer of electrons to carbon number 2, forming a ketone group at carbon number 2. And then the electrons within this double bond can attack the hydroxyl group at carbon number 5, which by doing so eventually the structure will fold into a 5-membered ring structure, which is the F6P. So that is the whole purpose of step 2. Nothing is gained, nothing is lost, but you have the rearrangement of the molecules. And then in step 3, there is an addition of another phosphate group to carbon number 1 to form fructose 1,6 bisphosphate. So the reason why it is called a bisphosphate but not a diphosphate is that in diphosphate or triphosphate, those phosphates are in touch with one another, just like in the case of ATP. But here, both phosphate groups, they are separated at carbon number 1 and carbon number 6. So it is going to be another energy requiring step where ATP is invested. This reaction is catalyzed by the enzyme phosphofructokinase, which is also known as PFK. So just for information, PFK is the enzyme that will set the pace of glycolysis because PFK is an allosteric enzyme that can be regulated by many metabolite binding. And again, you will need the presence of Mg2 positive as the cofactors of this reaction. Subsequently, a phosphate group is transferred from ATP to the carbon number 1 of this fructose 6-phosphate becoming fructose 1,6 bisphosphate. Now, if you look at the structure of this sugar, it is a 6-carbon sugar with 2 phosphate groups. And if you look even closer, it appears that this sugar looks like an adjoint twin similar to one another by the axis. Don't you think so? And indeed, fructose 1,6 bisphosphate will then be cleaved into 2 3-carbon sugar in the subsequent step. Okay, so we have done talking about the energy investment stage in glycolysis. Okay, here is a question to test your understanding. Please pause the video for a moment. And the answer is D, all options above. So in the very first step of glycolysis, you need the help from hexokinase, and you also need ATP, which will add a phosphate group to glucose producing G6P. For this enzyme to function, it will require the presence of a divalent ion, which is Mg2 positive as the cofactor. And how about this question? Well, the answer is B, to phosphorylate glucose molecule. So with the phosphorylation of the glucose molecule, it makes the metabolite less stabilized because they are now becoming more reactive, and therefore they are ready to join in the subsequent steps of glycolysis. And why is glucose phosphorylated? There is a second reason, which is very important. Okay, so the answer is B. As I mentioned just now, the main reason to make glucose phosphorylated is to make it unrecognizable by the glucose transporter so that they will remain inside the cell.